Efficient and accurate diagnosis of dengue and Zika is of primary importance for clinical care. Assalamualaikum and very good day everyone. Hi, my name is Dr. Siti Nur Khadijah Adis. In the previous video, you have learned about mosquito and human immune response against dengue and Zika infection. Now, in this video, I will be sharing with you the topic on laboratory diagnosis for dengue and Zika infection. This particular topic will cover first the current diagnostic method in dengue and Zika infection, and second, the advantages and limitations to current diagnostic methods. In this video, you will be learn about different laboratory techniques used to diagnose dengue and Zika in most laboratory settings. From that, you will be able to identify the types of sample use and advantages and limitations to each of the technique. In the end of this video session, you should be able to describe different types of laboratory diagnostic method in dengue and Zika infection, and you should be able to compare the advantages and limitations to each available diagnostic methods. A range of laboratory diagnostic methods has been developed to support patient management and disease control. The choice of diagnostic method depends on the purpose for which the testing is done, the type of laboratory facilities, the technical expertise available, cost, and the time of sample collection. Dengue and Zika can be diagnosed by isolation of the virus, by serological tests, or by molecular methods. Laboratory diagnosis methods for confirming dengue and Zika virus infection may involve detection of the virus, viral nucleic acid, antigen or antibodies, or a combination of this technique. These techniques are usually referred as direct detection methods and indirect detection methods. Isolation of virus by cell culture, genome detection by polymerase chain reaction or PCR, and antigen detection by enzyme-linked immunosorbent assay or ELISA are also known as direct methods, while the indirect methods usually detect the presence of IgM or IgG antibody through serological tests. Direct methods in detecting dengue and Zika are usually used in diagnosis of acute or recent infection during the first 5 days of symptoms. In virus isolation technique, virus is usually isolated from serum or autopsy tissue specimen and cultured either on mosquito or mammalian cells. Detection of specific dengue or Zika virus genome is now widely used through polymerase chain reaction or PCR. Sample from serum or plasma, cerebrospinal fluid, or autopsy tissue specimens during an acute febrile illness can be used in this technique with high accuracy and specificity. Finally, in direct methods, viral antigens such as the structural or non-structural proteins are used to indicate acute febrile illness through ELISA or dot blot assay. Dengue and Zika viruses can be grown in vivo, which is within the laboratory animal, or in vitro, which is the virus are grown outside of the living organism in cells in an artificial environment such as in a cell flask culture. Two most commonly used cells for isolation of dengue and Zika are Vero cells, which is a mammalian cells, and the C636 cells or Aedes albopicto larvae cells. Regardless of the method of cultivation, once the virus has been introduced into a host organism, or tissue culture cells, a sample can be prepared from the infected host or cells for further analysis under a bright field, electron or fluorescence microscope. Following infection in cell culture, the cytopathic effect or CPEs are usually observed. CPE are distinct observable cell abnormalities due to viral infection. CPEs can be seen as loss of adherence to the cell surface of the container, changes in the cell shape from flat to round, shrinkage of the nucleus, a complete cell lysis. Dengue and Zika virus can be detected in blood or serum from patient for approximately the first 5 days of symptoms through polymerase chain reaction or PCR. PCR is a technique used in the lab to make millions of copies of a particular section of DNA, and in this case, dengue and Zika nucleic acids. PCR was first developed in the 1980s. Currently, several PCR tests are employed to detect the viral genome in sample. That includes reverse transcriptase polymerase chain reaction and real-time polymerase chain reaction or RT-PCR. Since antibodies are detected later, RT-PCR has become a primary tool to detect virus early in the course of illness. Current tests are between 80 to 90% sensitive and more than 95% specific. In the process of PCR, a polymerase enzyme and thermal cycle machine are needed in order to produce multiple copies of viral DNA. In the antigen detection method, the virus non-structural protein 1 or NS1 and the structural protein has been shown to be useful as a tool for the diagnosis of acute infections. Dengue NS1 antigen has been detected in the serum of dengue virus infected patient as early as day 1 post onset of symptoms and up to day 18. The enzyme-linked immunosorbent assay or ELISA is a test that uses antibodies and color change to identify a substance. ELISA is a biochemistry assay that uses a solid phase enzyme immunoassay to detect the presence of a substance, usually an antigen, in a liquid sample or wet sample. Viral antigens from the samples are attached to a surface, then further specific antibody is applied over the surface so it can bind to the antigen. This antibody is linked to an enzyme, and in a final step, a substance containing enzyme substrate is added. The subsequent reaction produces a detectable signal, most commonly a color change in the substrate. On the other hand, a dot blot is a technique in molecular biology used to detect biomolecules and for detecting, analyzing and identifying dengue and Zika proteins. In a dot blot mixture containing the viral antigen to be detected is applied directly on the membrane as a dot and then is spotted through circular templates directly onto the membrane. This is then followed by detection by either nucleotide probes or antibodies specific against dengue and Zika. Indirect methods in detecting dengue and Zika are becoming an important tool for routine diagnosis as it has a high specificity and sensitivity, however, only when used at later days of infection. The MAC ELISA or IgM ELISA and IgG ELISA detect the presence of specific antibody against dengue and Zika, which detect both primary and secondary infection. Hemagglutination inhibition assay is an indirect approach used to detect the presence of certain types of dengue or Zika in patient serum. Hemagglutination inhibition assay can be used to detect the presence of antibodies specific to viruses in a patient even months or years after infection. IgM antibody capture ELISA and IgG ELISA are most commonly employed in diagnostic laboratories and commercially available diagnostic kits. In ELISA, the total IgM in patient serum is captured by anti-mu chain-specific antibodies, which is specific to human IgM, coated onto a microplate. Dengue or Zika specific antigens are bound to the captured anti-dengue or anti-Zika IgM antibodies and are detected by monoclonal or polyclonal dengue or Zika antibodies directly or indirectly conjugated with an enzyme that will transform a non-colored substrate into a colored product. The optical density is measured by spectrophotometer. The IgG ELISA used for the detection of a past dengue infection utilizes the same viral antigen as the IgM ELISA. Hemagglutination assay is a serological assay used to detect the presence of certain types of viruses in patient serum. Serum is a liquid fraction of blood plasma from which clotting factors have been removed. Serum can be used in hemagglutination assay to detect specific types of viruses in the patient's sample. Hemagglutination is the clumping together of red blood cells. Many viruses produce surface proteins called hemagglutinins that can bind to receptors on the membrane of red blood cells and cause the cells to clump. The specific antivirus can be used to bind to components such as hemagglutinins that are uniquely associated with specific types of viruses. The binding of the antibodies with the hemagglutinins found on virus subsequently prevent erythrocytes or RBC from directly interacting with the virus. So, when erythrocytes are added to the antibody-coated viruses, there is no appearance of agglutination, in which the agglutination has been inhibited. The acquired immune response following a dengue infection consists of the production of IgM and IgG antibodies. The immune response varies depending on whether the individual has a primary, which is the first dengue or other flavivirus infection, versus a secondary, which is had dengue or other flavivirus infection in past. The primary versus secondary dengue infection can be determined by using a simple algorithm. A primary dengue infection is characterized by a slow and low titer antibody response. IgM antibody is the first immunoglobulin isotype to appear. Anti-dengue IgG is detectable at low titer at the end of the first week of illness and slowly increases. In contrast, during a secondary infection, antibody titers rise extremely rapid and antibody reacts broadly with many flaviviruses. The high level of IgG are detectable even in the acute phase, and they rise dramatically over preceding 2 weeks. After you have learned about some basic principles in laboratory diagnosis for dengue and Zika infection, let's see about the advantages and limitations of each of the diagnostic methods for each purpose. As seen on the table, each of the available methods comes with various advantages and limitations. Can you determine which method has high sensitivity and specificity? Can you determine which method is less expensive and easy to perform? Can you identify which method is able to distinguish between primary and secondary infection? Now, having watched this video, I hope you can now describe different types of laboratory diagnostic methods in dengue and Zika infection, and be able to compare the advantages and limitations to each available diagnostic methods. I hope you have enjoyed this video and slideshows and have gained knowledge on laboratory diagnosis for dengue and Zika. In the next video, you are going to explore another interesting topic, which is treatment and myth about dengue and Zika. Thank you.